you have all these flasks, and in them they should have bacteria that should have your protein in them that you wanted them to make. But what if you go to purify them and you can't find that protein? What do you do then? Well, sometimes proteins don't express well in bacteria, and sometimes they express well, but they don't fold properly, and then they get clumped together in aggregates called inclusion bodies. So you can do things like run gels to the different portions, like the soluble portion, the membrane bits, and that sort of thing, to see if your protein is getting expressed and it's ending up in inclusion bodies, or it's not getting expressed at all. And depending on what the problem is, there are different things that you can try to do to get the protein to express better. These involve things like changing the cells and changing the protein in simple ways. So let's take a look at how we can do this. Yesterday we talked about when things go right with bacterial protein expression. So that's where we get bacteria to make a protein that we're interested in. But say you do that and it doesn't work out. That's actually pretty common. So let's talk about some of the things that you can do to try to maximize your chances of success and at least figure out what's going wrong and try to fix it. So something to start with is that not all proteins are going to express well in bacteria. It's just part of the game. You're going to have the most luck if your protein is small and it doesn't have a lot of modifications like phosphorylation or glycosylation, so the addition of sugar chains. So bacteria cells have the ability to read the genetic code, and they have the ribosomes to make the protein, but they don't have all the same machinery as like our cells. So they don't have the ability to do all the same modifications in the same way. So they can do some modifications, but they might do them differently. They might not have the chaperones, so the folding proteins that help the proteins fold properly. So without those, the proteins might not express well, or they'll express and then they will aggregate, so they'll clump up, and then you won't be able to purify them. So that's the basics of the types of problems that you might face. So sometimes you might have better luck, especially if your protein is big and modified, with going to a different cell line, like insect cells or mammalian cells. But that can be a lot more expensive and require a whole more time and stuff. But assuming that your protein is capable of expressing in E. coli, there are things that you can do to increase your chances of success. So let's talk about some of those. Don't worry about all this text for now. We'll go through it. I just wanted to give you an overview of what the type of things we're going to be talking about. So I'm going to assume that you know some basics about recombinant protein expression. So recombinant meaning we recombine DNA, so we stick the genetic instructions for a protein of our interest into DNA, a DNA vector, which is going to serve as a vehicle for sticking that DNA into cells that will express the protein for us. So I have a lot more about this in previous videos, so I'm not going to go into it now. But so what we're going to talk about today is mostly for bacterial protein expression, although a lot of the things we're going to talk about are applicable through any expression system. So one of the things with the expression is, so with bacteria, you're typically sticking it in a plasmid, which is a circular piece of DNA. So you can stick in different genetic recipes, or which we call cDNAs, complementary DNAs, which are DNA versions of the mRNA. So more yesterday's post. So you can stick in different cDNAs to get different proteins, and you're controlling the gene, so you can stick extra bits on it, like stick on some extra amino acids, so extra protein letters on the end, which will allow you to serve as a tag to allow you to grab the protein out more easily with affinity chromatography. So chromatography is basically where you send your protein
a protein mixture traveling through a column filled with little beads called resin, and these beads have different properties that are going to separate the proteins based on the protein's properties, like charge, size, that or presence of a tag, that sort of thing. So with today we're going to talk about what happens when you do that and you end up with nothing. So there's a lot of places that things can go wrong. So say you're not checking anything, and then you go through that affinity chromatography step, and such as like a his tag purification or a strep tag purification, and you don't have anything. So there are a lot of places where things could have gone wrong. So we can break up the recombinant protein expression workflow into a few main stages. So there's the molecular cloning phase, where you're sticking the gene of interest or the cDNA of interest into that vector. Then there's the expression stage, when you're actually getting the protein expressed. And then there's the purification stage, where you're breaking open the cells and trying to purify out the protein. And you can have problems at any stage. And there are different ways we can test for those different problems. And based on where the problem is, there are different things that you can try to counteract the problem. I'm going to talk more, but so basically this is showing it from our point of view kind of thing. But after the expression stage is kind of complicated, because there's the actual expression. So there's the making of the, there's the transcription of the DNA into the mRNA, and then there's the mRNA making into the protein. But then before you can actually get to the purification stage, there's the problem of the protein might actually express, but it gets like it misfolds and gets aggregates or clumps up into these things called inclusion bodies, which are just like clumps of protein inside the bacterial cells. And the protein could also get degraded and that sort of thing. So there are things that can happen after the actual physical expression that prevent you from being able to actually purify out the protein. And there are also problems with the purification itself, like if your resin, you use the wrong resin, or you accidentally wash your column with the competitor instead of the wash buffer, or the resin's gone bad, or that sort of thing. So there's lots of different places that things can go wrong. So as we talked about in yesterday's video extensively, with bacteria we're often using an overexpression system, so an inducible system, where the bacteria, the gene of our interest, is under the control of this thing called a T7 promoter. So T7 is a bacteriophage, so a virus that infects bacteria. And it has its own RNA polymerase. So RNA polymerase is what transcribes the gene into mRNA, and then that mRNA gets translated into protein. So the more you transcribe the gene, the more mRNA you have, and the more protein that you can make. So I'm not going to go into too much detail, as I said, because I talked about this extensively yesterday. But the idea is that when you add this molecule called IPTG, you can induce the T7 polymerase to be made, and then so your mRNA is made and you get your protein. So the way it's done is in these big flasks usually. You let the bacteria grow to get a large workforce of bacteria, basically. So you want to have lots of bacteria. Then when you add the IPTG, the bacteria stop growing and that sort of thing, and instead focus on almost entirely on making your protein. So you can end up with about half of the protein being yours. So it's pretty awesome. But sometimes if your protein is harmful to the cell or doesn't fold well or that sort of thing, if you try to express too much, then the protein is going to aggregate, or it's not going to have time to fold properly or that sort of thing. So it's kind of like you're making a lot, but it's really low quality because you're going too fast, or you're just overflowing your cells with this protein that isn't very helpful to them. So sometimes you actually, one of the things that you can do if you're having expression problems is use a lower IPTG concentration, or don't let the expression go for as long, or lower the expression temperature. So once you express the protein, then you lyse it, and so you break open the cells and you spin it down, and so you separate the soluble parts from the non-soluble parts, and then you do the chromatography where you send it through those different resins that are going to separate the proteins based on their properties. So we're going to come back to this later, but when you do this ultracentrifugation step, so you remember you can also just use like a normal centrifuge, but going really fast as you can. If you don't have this ultracentrifuge available, but basically the membrane bits and are usually going to be in your pellet. So this is going to be like the non-water soluble stuff. So we're going to be assuming throughout this video that you're trying to purify a water soluble protein. So just like a normal protein that hangs out inside in the cytoplasm, so the general interior of the cell, and not like a membrane bound protein, which you would expect to find in the pellet. So you're not expecting to find your protein in the pellet, but sometimes it can be. So if your protein is aggregating and going into those inclusion bodies, it's going to end up in the membrane bit. And so when you run a gel, if you're not seeing expression, you should test that membrane fraction. So take a little bit of that pellet, mix it in a bunch of buffer, SDS loading buffer, boil it, spin it, and take a little bit of that and see if the protein is there, because that'll tell you it was expressed, but it's going into this insoluble portion. If that happens to you, there are things that you can do to try to purify it out of the inclusion bodies and refold it. I've never actually done that, but it is possible for some proteins, but you have to be sure that they're refolding properly. So ideally you'll have some sort of functional assay, so some sort of readout of whether the protein's working. So say you're purifying an restriction enzyme, so an enzyme that cuts DNA. And if you have so you can test and see, does it cut DNA? If it does, there's good chances that your protein is made properly. If you don't have a sort of functional assay, you can do with size exclusion chromatography, you should see a nice sharp peak representing a well-folded protein at least. So with any purification, so assuming that you're not in the inclusion bodies, what you want to do, so the first step is to make sure that your protein is actually being expressed. So to do this, so typically what you do is you take a pre-induction sample. So you take a bit of that bacterial culture before you add the IPTG. And then you take a sample before you harvest the cells, so before you go to spin them down, but after you've let them express the protein. Since you're overexpressing the protein, you should be able to see the presence of a new band at the size that you're expecting in your post-induction sample. So as you can see here, there's this nice band that's in the post-induction sample, but not in the pre-induction sample. So that's telling me that the protein was expressed. Yay! But this is telling me, so this part is actually, this is before we do the purification. So this, I don't even know if it's in the soluble portion, but at least I know it was expressed. So what happens if you don't see that big fat band? So is your protein being expressed at low levels and you just can't tell it apart from the background, or is it not being expressed at all? And so then one of the things you might want to do is a Western blot. So the basic idea with a Western blot is, so what I was showing you before was a Kamasi. So basically it stains all the proteins. What you're going to do with a Western blot is you use antibodies to look for specific proteins. So this is an SDS PAGE gel. We're separating proteins by their length in this gel mesh. And so with the Western blot, you're actually using antibodies, so little proteins that are specific for the protein of interest. And then you can see if the protein is present. So you should see that protein present in your post-induction sample, but not your pre-induction sample. Or at least in very low levels in the pre-induction sample. So ideally, that's the case. Sometimes antibodies are not very good. So you'll want to have like a positive control. So a protein that you know should react to the antibody. So this could be a problem if you don't have a pure version of your protein. But if your protein, say you have a his tag protein, there are antibodies for the his tags. So instead of using antibody specific for your protein, you can use antibody specific for the tag, and then you'll probably have a protein with that same tag in your lab, hopefully, that you can then use to as a positive control. So say your protein is not expressing. What you want to do, there's a few things. So first you want to make sure that the cloning is all good. So you take, when you're doing the cloning step, typically, you start out in like cells that don't have T7 polymerase. So these are not expression cells. These are just like cloning cells that you can use to make lots of copies of the plasmid. So they're like quick tests you can do in the lab to see if your insert probably got inside, like colony PCR and analytical digest. But those won't tell you if the sequence is correct. So to really know if the sequence is correct, you need to send them for sequencing. So when you're using sequencing, basically you'll use primers that are going to copy into the inserted gene, and then you're going to send them for sequencing, and you'll get back these wavy line things representing the sequence that you're then going to compare with the sequence that you expect to see. And you're looking for conflicts where there are any mutations that are like differences between your sequence and the sequence that you want. You especially want to be careful about the ends, which is like, so where you cut and pasted it in or slicked it in or whatever, and you want to make sure that the ends are all good. So if your clone is okay, you want to make sure that the cells that you're using for expression and stuff are okay too. So you want to hopefully you use controls when you were doing your cloning, so to make sure that the cells were competent so they could actually take in that plasmid. So assuming that your plasmid is okay, the cells are okay, so now the problem seems to be something with either the expression or post-expression stability. So now we need to talk a little bit more about what makes proteins and what makes them stable and how they form and fold and what can go wrong. So proteins are basically long strings of these letters called amino acids. Those strings, amino acids, have this generic backbone that allows them to connect up together, and then they have these side chains or R groups that are the unique parts, and they hang off from the chain. They're kind of like charms in a charm bracelet, and they have different properties like size and charge that affect how the protein is going to fold up. So the fold chain folds up in a way that makes all of those side chains as happy as possible. So you want to stick negative charges next to positive charges. You want to stick the hydrophilic things, so the things that water likes, on the outside by the water, and the hydrophobic things which the water excludes in the center. So this gives you this 3D protein, which is functional. So a little more about those hydrophilic and those hydrophobic terms, because that comes up a lot with the protein solubility. So it's easiest to think about protein folding in terms of like positive charges when it goes to negative charges and that sort of thing. But the main driving force of protein folding is actually this hydrophobic effect. So basically water is highly polar. So what this means is that even though water is neutral overall, so it's uncharged overall, it has positive parts of it and negative parts of it, like partly positive and partly negative. So it's beyond the scope of this post really, but atoms are made up of protons, neutrons, and electrons. Protons are in the center and they're positively charged, and electrons are around them and they're negatively charged. And the electrons are in this cloud, so you can think of this cloud shifting, so sometimes it can hang out more with one part of the molecule, sometimes more in the other part of the molecule, And the oxygen atom is really electronegative, so it's an electron hog, so the cloud of electrons is going to hang out more with the oxygen, and so you have more negativity around the oxygen and more positivity around the hydrogens. And so now you have partial positive, partial negative. Now you can get to your opposites attract, and so water really likes to stick to itself and to other partly or fully charged things. So if you have things that aren't charged at all, so you have hydrophobic things, the water is going to exclude those things. So the water is going to try to minimize its contact with those things because the water is trying to maximize its contact with things it likes. And so the things it doesn't like are going to get kind of clumped together. So the hydrophobic things gather together. And so this is a key force of protein folding, because the hydrophobic parts of proteins are going to fold together, kind of like huddle together in the core of the protein. But if you don't allow the protein time to fold properly, or you don't have, sometimes proteins need like chaperones, which kind of help the protein fold properly. So they protect the hydrophobic parts before the rest of the protein comes out of the ribosome, so that they can, so it's kind of like a waiting, it can serve as kind of like a waiting room. So if the part of the protein comes out that needs to bind to part of the protein that hasn't been made yet, then it can kind of protect it until then. So that's one of the ways chaperones can help out. But if you don't have those chaperones, or if the protein you're trying to make it too fast or that sort of thing, the protein might misfold, and then those hydrophobic, the wrong hydrophobic parts might clump together. Or if the protein misfolds and the hydrophobic parts end up on the outside of the protein, then they're gonna clump to other hydrophobic misfolded proteins, and that's how you end up with things like inclusion bodies, so those big aggregate clumps. So your goal is a soluble protein, and so what we mean by soluble is that each copy of the protein has this full water coat. And so as I talked about, you have this protein called aggregates, or these problems where your protein forms aggregates. And so one of the solutions that you might want to try is, so we talked about how you could try lowering the IPTG concentration, decreasing the expression temperature. So instead of expressing it at like 37 degrees Celsius for a few hours, you can express it for at like 18 degrees Celsius overnight. You can also reduce the expression time. So those are things you could try to make less of the protein. You could also try different expression cells. Another thing you can try to do is, instead of changing the bacteria, change the protein. So you don't have to change the whole protein, just add a little bit on to the end. So we often use these things called fusion proteins, which are little proteins that are very soluble. They fold really quickly and easily, and the cells really like making them. And so you stick one of these onto the end of your protein. And it's kind of like a foot in the door campaign in a way. So you have this fusion partner on the end of your protein. The bacteria makes the fusion partner, and then it goes right on to making your protein. So since it really likes making that fusion partner, you kind of like trick it into making your protein. And if you put a protease recognition site, so like a cut site in between them, you can then cut that tag off when you're done with it. And in addition to this, these fusion partners can have multiple effects. So they can also serve kind of like a chaperone, like protecting the ends of your protein, as well as that foot in the door technique. And kind of shepherding your protein throughout the cell to more protected areas. Another thing you can do is actually to change the sequence of your protein without actually changing the protein. It's something called codon optimization. So the basics of translations to the process of protein making is that the genetic instructions for the protein are written in mRNA. So three RNA letters, so that we call that a codon, and it spells one amino acid. And so the ribosome travels along the mRNA, and it's inserting the amino acid that corresponds to the codon that it stopped on. And so this is where you can see like one end is coming out and the rest of the protein hasn't been made yet. And so that's where the chaperones can really play a role in helping protect those ends. And this is why if you put the fusion partner on the N terminus, then it kind of tricks the cells into making your protein. But what I want to talk about here is that, so you have those 3-letter codons, but there are actually multiple codons that can spell the same amino acid often. And so if you cells tend to stock up more on the tRNA. So the tRNA is the molecule that brings the amino acid. So on one end it has this anticodon that matches the codon, and then on the other end it holds on to the corresponding amino acid. And so different tRNAs recognize different codons, and so you can have multiple tRNAs for the same amino acid that are recognizing multiple codons. So cells tend to have like codons that they prefer, which is called codon usage bias, and so they're going to stock up more on those corresponding tRNAs. So if your sequence has the a lot of rare codons for that cell type, so if the spelling, there are 6 codons that spell leucine. E. coli uses CUG the most and CUA the least. So if the gene you're trying to put in there has a bunch of CUA, then the bacterial cells aren't going to have as much of that tRNA in stock, and then the protein making might stall. So you can use codon optimization to change those CUAs to CUGs and hopefully get better expression. Of course, that can be, so you can send it to companies and stuff that can do this, but this can be expensive, and so an alternative is to change the bacteria. So use something like a Rosetta strain of E. coli that has extra copies of the rare codons, the rare tRNAs. So change the bacteria's like tRNA composition to help. Okay, so basically in an overview, we went through a lot today. So sorry it went long. I just wanted to give lots of details for those who wanted them. So basically, the idea is to make sure you check your pellet. So see if your protein isn't expressing. First of all, you try to make sure it is expressing. So you're going to do the SDS page. You're going to do the Western blot. You're going to check the pellet, check the supernatant. If your protein is not expressing, then you can do things like lowering the temperature, decreasing the IPTG, adding fusion. If it's expressing, and if it's expressing like but misfolding or whatever, so basically you can. So if it's not expressing at all, you might want to try like optimizing the codons, that sort of thing. Make truncations to cut off predicted disordered regions, or add the fusion partners if it's. So you might want to try, if nothing works, you want to try a different expression system like insect cells or mammalian cells instead of bacteria, or different bacterial strains. We didn't even talk about, but there's also the potential that the problem was with the purification stage. And so to test for that, so you want to make sure that you're taking samples at like all the steps along your protein purification process. So if you have evidence that your protein expressed, you have evidence that it's in the soluble portion, you're just not getting anything off of your column, you want to check that you're doing everything right with the purification. So you want to be taking steps, taking samples of your input onto the column, the flow through, so what's going through without you adding the competitor, the wash, so what happens when you add the buffer without the competitor, and then the elution, so what happens when you add the competitor. And so if you had protein and then you try to purify it and you don't have anything, the protein should still be somewhere. So you want to try to figure out where it was. And then if there was like a problem with the resin, so with the little beads, or maybe you accidentally added the competitor when you didn't mean to, when you were thinking you were doing a wash and you were really doing an elution, which I've done before and it's very, very painful, then that would show up there. And so basically the goal is to figure out what stage there's a problem at, and then hopefully you can fix that problem. So that's just a sort of whirlwind overview of things that you can try, and I hope that helps someone. And sorry if I just really bored someone, but I'm guessing if I really bored you, you're probably not here still. So happy purifying and best of luck.